It is a meeting not just of management committee members, but anyone else who has been involved with hCOMET over the years has been invited. So a special welcome to all of you. So I'm going to talk about the background to hCOMET and give a summary of some of the activities that we've been involved in over the past 4.5 years. The origin of hCOMET is actually in another project, hCOMET, which we launched probably nearly 10 years ago. And the aim of hCOMET was to collect comet assay data from as many human trials as we could, and to create a common database so that we could carry out a pooled analysis. And the aim of hCOMET really was to find definitive answers to questions that have bothered us over the years, simply because different labs have come up with results that are sometimes inconsistent. So doing a pooled analysis should tell us what the real facts are. Questions such as, does DNA damage increase with age? Are there differences in DNA damage between men and women, between smokers and non-smokers? And what are the effects of exposure to various environmental and occupational mutagens? The model for the pooled analysis that we planned was the work done on chromosome aberrations and micronuclei, which had demonstrated these to be predictive markers of cancer risk. In other words, if you had a high level of chromosome aberrations or micronuclei at a young age, that meant that you had a significantly elevated risk of cancer later on. So there's a further question that we can address to COMET assay results. Is DNA damage a biomarker of cancer risk? Maybe you think this is likely. I was fairly skeptical because, for example, there's a lot of evidence that DNA damage is elevated in cancer patients, but this, of course, could be an effect rather than a cause of cancer. And DNA damage is an initiating event in carcinogenesis, but remember that almost all the DNA damage is repaired, and even the unrepaired DNA damage is unlikely to cause mutations in cancer genes. So the link with cancer is really quite tenuous. To sort out this problem, really we need long-term cohort studies, either prospective studies or retrospective studies, and you'll hear about this more about this later. hCOMET was unfunded, but even so, we managed to attract over 100 researchers to join us, and half of them undertook to provide us with their COMET assay data. And then we successfully applied for a cost action to carry out the aims of hCOMET, and also an additional aim to attempt to standardize the COMET assay in order to reduce the variability between labs and to make inter-laboratory comparisons easier and more reliable. So this 4-year action was launched in April 2016. Officially, it ended in April this year, but because of COVID-19 pandemic, it was extended by 6 months to allow us to have our final meetings in Croatia. Unfortunately, of course, that also was unable to take place, and instead we're having this online meeting. We have 25 member countries in hCOMET. The number has increased over the years, and they cover most of Europe, as you can see, with some notable exceptions, but we have a pretty good coverage. And I think it's important to note that more than half of the countries are inclusiveness target countries. These are countries where scientific endeavors, according to the EC, can be improved and brought up to the level of science and research in other countries. We also have observers from near neighbor countries, Jordan and Kosovo, and I hope the members from there are attending this meeting, and also from international partner countries, India, Cuba, Brazil, and USA. So a special welcome to them. hCOMET is organized around 7 working groups. The first and I suppose principal working group is tasked with creating and analyzing this enormous combined database. The second working group deals with technical innovations and improvements in the assay and making sure that these can be implemented. Then we have ring studies, the aim of which is to carry out experiments using a standard protocol in different labs to see if we can reduce the variability between labs and come up with standard procedures, which are the topic of working group 4, producing guidelines and SOPs. One of the most important applications of the COMET assay is in studying DNA repair. And so we have a working group dealing with DNA repair assays. Working group 6 deals with the use of different cell types, principally the different cell types that are used as samples in human trials, but also looking at human cell lines. And then an important working group for the future is number 7, planning how we can go forward from here. So you'll hear about the progress made in each of the working groups in the presentations that follow. As I said, the primary aim of hCOMET was and is the creation and analysis of a combined database. And we've received almost 20,000 individual sets of data on strand breaks, enzyme sensitive sites, hydrogen peroxide sensitivity, which is a measure of antioxidant resistance, and DNA repair capacity. And these data have come from 108 human trials and 38 research groups. Analysis, as you can imagine, was a mammoth task. And you will hear about some, I think, very interesting and exciting findings. As I said earlier, we have this important question whether DNA damage is a biomarker for cancer risk. And to investigate this possible link, we need a large-scale, long-term prospective study, or, as an alternative, a retrospective nested case control study. And this could be done using samples from an existing long-term, large-scale trial such as EPIC, the European Prospective Investigation into Cancer and Nutrition. And we know that EPIC has stored frozen buffy coat samples. These are blood samples which are rich in white cells. And we know, as you'll hear later, that the white cells in buffy coats can be analyzed using the COMET assay. I won't say more about that now, but you will hear more later on. The second aim of hCOMET is to deal with COMET assay variability and hopefully to reduce that variability. For many years now, we've been concerned with the variations that are seen in results coming from different labs. And this has been highlighted in various collaborative studies, especially hCOMET and ICAWG over the past 2 or 3 decades. And these differences can arise from technical factors, and it's clear that many labs use their own version of the COMET assay. They make variations in the technique, and sometimes this is done without a proper understanding of the underlying principles of the assay. So in hCOMET, we've emphasized the influence of specific practical aspects on the results. For example, the agarose concentration is important. The higher the concentration, the more difficult it is for DNA to migrate. Lysis period can be important depending on the type of DNA damaging agent involved. If we're using the enzyme modification of the assay, it's important to make sure that we have the optimum conditions for the enzyme digestion. The time spent in alkali can be important, if only because it converts alkali labile sites, such as baseless sugars, into breaks. And then electrophoresis is really important, and it can be affected by, or the migration of the DNA can be affected by the time of electrophoresis and by the voltage gradient. Finally, the scoring method. It's been shown particularly in the ICAWG trials, that scoring varies a lot between labs, and that can account for a lot of the variability. So we've been putting a lot of effort into developing standard operating procedures in the hope that these will be adopted by many labs, and this will reduce variability and make it much easier and more reliable to carry out inter-laboratory comparisons and collaborations. A very important objective of all cost actions, and hCOMET, of course, is the creation of skilled young scientists. And we have been working hard to provide skills for a cohort of young molecular epidemiologists in the future, providing training courses and supporting exchange visits between labs. We've had several very successful training courses. The first was a course on how to measure DNA damage and DNA repair with the COMET assay, basic techniques, in Oslo. We then had a course in Rome on statistical and epidemiological issues in human biomonitoring studies. There was a course on the use of different cell types in Riga, a rather specialized course on the COMET assay applied to 3D cell systems in Oslo. And the last course was, again, a course for beginners in the technique, explaining basic COMET assay techniques, and that was in Sarajevo. Short-term scientific missions are an excellent way of using funding from the cost action to support real science, and we've managed to support a total of 32 short-term scientific missions. So these have provided training in specific techniques for the most part, young researchers, as well as valuable experience of working in another lab in another country. But they've also contributed significantly to the scientific output of hCOMET. And then there are ITC conference grants. Early career investigators and PhD students from these inclusiveness target countries have been eligible for special grants to attend conferences. And we were able to support 26 young scientists in this way. Publications, I think our record is fairly impressive, with over 70 papers published with hCOMET authors, including more than 40 which have co-authors from 2 or more hCOMET partner countries. We were commissioned to produce a special issue of Mutation Research, which actually turned into 2 special issues, one on the COMET assay in human biomonitoring and one on the in vitro COMET assay. And recently, we've had accepted 2 papers of protocols in the journal Nature Protocols, and another one is under in preparation. We also made videos, one on goals and achievements of hCOMET, another on methods for isolating white blood cells, and a third on the COMET assay applied to DNA repair. And there are others in the pipeline. The website is going to continue to exist after hCOMET, and it'll provide access to videos, details of publications, guidelines and SOPs, and also an innovative catalogue of human cell types and cell lines, which you will be able to refer to and contribute to. It'll also have news of events and developments in the Common Assay world. So I'd like to thank everyone who's taken part in this, I think, very successful and enjoyable cost action. And so now we'll carry on with the presentations from the various working groups. But if there are any questions, I'll be happy to answer them.
Stefano.
Only to say thank you very much, Andrew, because you are a wonderful coordinator. You were always present from a scientific and practical point of view. You were really close to everybody. When we had problems, you were there. And so I know that is a period in which you changed your life, you retired, but you were still present. And I think that I really want to thank you on the name of everybody for your work of these 4 years.
Thank you. I'm touched by that, Stefano. I've really thoroughly enjoyed it, and I think there have been a lot of friendships made, and I'm sure they will carry on forever. Well, as long as COVID-19 lets us live. Well, thank you, everyone.